Hey everybody, welcome to General Biochemistry Lecture. Today we're going to talk about Chapter 2 in Linear Principles of Biochemistry. The chapter is about water, which is the solvent of life. These are the key principles that you should take away from this chapter. At the end of this chapter, you should be able to come back to this slide and 1, understand what each of these points is discussing, and 2, be able to expand upon it. These are the topics that you'll need to understand and be able to do problems on when it comes time for quizzes and exams. We're going to start with weak interactions in aqueous systems. We're going to talk about water. Before we start talking about the properties of water, we need to discuss the molecule. So water is H2O. And the bonds between the oxygen and the hydrogen are covalent. Covalent bonds are strong. There's a pretty big difference in electronegativity between oxygen atoms and hydrogen atoms. So you'll notice this lowercase delta with the plus sign near the hydrogen atoms, and the delta with the minus sign for the lone pairs on the oxygen. That means partial positive and partial negative. So water is made of hydrogen and oxygen. They're covalently bonded, and it is a polar molecule, meaning that the electrons are not shared equally between the oxygen and the hydrogen. The shape of the molecule, if you remember Vesper theory, the shape of the molecule is dependent upon the atoms, their electronegativity, how those electrons are shared, and how the lone pairs impact the shape as well. So the shape for water is bent. So that's just a little bit of a general chemistry refresher on how to look at molecules. Now we'll talk about the unusual properties that water has. It's got a high melting point, boiling point, and heat of vaporization that's higher than most other common solvents. And that is due to hydrogen bonding. But the hydrogen bonding that we're talking about is not within the molecule itself. We're talking about 2 molecules of water, like in figure B here. We've got molecule 1 and molecule 2. The oxygen from molecule 1 is attracted to the hydrogen in molecule 2. That is a hydrogen bond. It's the electrostatic attraction between oxygen and one water molecule and the hydrogen of another. You'll notice that the distance between these 2 molecules is much greater than the distance between the hydrogen and oxygen within a water molecule. So that covalent bond is short and strong. Hydrogen bond is longer and weaker. Speaking of strength, like I just said, hydrogen bonds are relatively weak. Okay, they're mostly electrostatic, and they're very, very fleeting. Now, you don't need to know the numbers on this slide, but I do want you to know, I'll highlight it here because I can, that hydrogen bonds are relatively weak, that they're mostly electrostatic in nature, and that they are very fleeting. When one hydrogen bond breaks, another one forms. So you definitely want to know that. Now, you don't need to know the numbers on this slide either, but I do want you to understand when you have liquid water, each water molecule forms hydrogen bonds with, on average, about 3.4 other molecules. And in ice, you're forming bonds with 4 other waters. So you have this structure of water where these hydrogen bonds are constantly forming and breaking, forming and breaking. It's very dynamic. It'll change the way you look at water next time. So water forms hydrogen bonds. It can form hydrogen bonds with itself and with other molecules. So the way that I was taught about hydrogen bonds, which is a little bit silly, but I've remembered it all these years. Fawn with hydrogen bonds. Fawn stands for fluorine, oxygen, and nitrogen. And those are the three atoms that you can have a hydrogen bond with. Now, for our purposes in this class, we're not really talking about fluorine. But oxygen and nitrogen are very biologically relevant. So we're focusing on those hydrogen bonds. The hydrogen acceptor is always going to be the electronegative atom. So in this case, the oxygen or the nitrogen. The donor is always going to be a hydrogen from water, or it could be attached to another molecule or another atom like a nitrogen. We're going to talk about some of the different macromolecules that participate in hydrogen bonding. So you'll notice a lot of carbons with hydrogen atoms. Those do not participate in hydrogen bonding. However, if you see hydrogens that are a part of an alcohol, or if you're looking at a ketone, if you're looking at compounds that have nitrogens with hydrogens attached to them, those are all fair game for hydrogen bonds. So you'll need to recognize places where hydrogen bonding can occur. So you can already see it here, but I'm going to just go through and highlight just to show you. There's a hydrogen bond there, there, here, and here. So on an exam or a quiz or an assignment, you'll need to be able to look at two molecules and say, oh, okay, there can be hydrogen bonding here, there can be hydrogen bonding there, there can't be hydrogen bonding there, that's attached to a carbon. So water interacts with a lot of different things, okay? And it interacts electrostatically with charged solutes. So what does that mean? What it means is if you have a salt or some kind of polar compound, then that water is going to be able to dissolve it, and it's going to interact with the different charged ions or the different areas of the polar molecule. So remember, polar means partial positive, partial negative. We've got some other terms that you'll need to know here too. We've got hydrophilic, and that just describes something that's easily dissolved in water. Hydrophobic, you'll need to know that. Nonpolar, so lipids, waxes. And amphipathic. That means that it's got both a polar or a charged region, and it's got some nonpolar regions in there too. And we'll give examples of all three of those. So for this table, I don't want you to necessarily memorize these structures, but I want you to look at a molecule and be able to recognize when a group is polar or nonpolar. So this is kind of drawing from your organic chemistry brain. And if that's a little bit of struggle, then you will definitely want to review your functional groups, because when we get to chapter 2, we're in chapter 2. When we're in chapter 3, we're definitely going to be steamrolling through and looking at lots of structures, and you're going to need to recognize those functional groups. So word to the wise, dust that memory off. So let's talk about water as a solvent. It is the solvent of life, right? Water dissolves salts and other charged molecules pretty much by giving those molecules something else to interact with. So let's say that you're about to boil some pasta and you have some water, it's boiling, you add some salt. The salt dissolves, you just give it a stir, bam, you don't see it anymore. What happens? Well, when you add that salt, it's initially a crystal, and that's what's shown here. Okay. And when it's added to the water gives the chlorine ions and the sodium ions something else to interact with. The water says, hey, chloride, I've got some positive charge that you can interact with. So I'm just gonna, orient my hydrogens towards you because I know you like positive things. And the opposite happens with the sodium. Hey, sodium ion, you're very positive, and we're not trying to deflate your balloon or anything, but we know that you like to interact with that negative charge. So here's some oxygen. And then slowly that crystal breaks apart. This is very favorable because it increases the entropy of the whole system. And remember that entropy is just the fancy scientist word for chaos. So we talked about charged stuff. Now we're going to address some nonpolar things. There are some gases that are really important for biology. Carbon dioxide, oxygen gas, nitrogen gas. Highly important, also nonpolar. That means that they don't dissolve well in water. But why? If you think about gases, when you learned about it in general chemistry, if I had a box of gas, right? Those are my gas molecules. They're going to be zipping all over the place, bumping into each other, bumping into the walls. Free as a bird, enjoying life. You constrict that movement into an aqueous solution, you're decreasing the entropy. So you're ordering the gas more than it wants to be ordered. And that is not favorable. So nonpolar gases, not very soluble in water at all. And you can see that in this chart here where the solubility in water in grams per liter, very, very small in comparison to something like ammonia, which is a polar molecule. You can dissolve a lot of that. Amphipathic compounds. Really, when we say amphipathic, the first thing that should come to your mind, well, it will after this chapter, is lipids. Lipids, cell membranes. You have a polar region or a charged region, and you have a nonpolar region. The polar region, if we're talking about some kind of a fatty acid, will have a hydrophilic head group, which is here. Then you'll have the lipid tail, which is all of this craziness, the yellow. And let's say that you're trying to make some kind of a dilute solution of a lipid. Well, you're going to have ordering of water molecules around that nonpolar part, but not actually interacting. So in your book, it calls it these cages around the hydrophobic tails. But you will have water interacting with that hydrophilic head group. Now, let's say that you have a high enough concentration to where the lipids can start to cluster together. That's called the hydrophobic effect, where those nonpolar regions come together so that they can interact with each other, and the charged or polar regions can also interact with each other and with the water around it. Eventually, if you keep increasing the concentration of those lipids, you'll form what's called a micelle. And that's literally just like a ball of lipid where you've got your charged or your polar head groups on the outside, and you've got your non-polar lipid tails on the inside interacting with each other away from the water. There's one more interaction type that we're going to talk about. That's van der Waals. All of these terms may be a little bit like, I feel like I've heard that before, and that's good. These are all intermolecular forces that aid in keeping big molecules together. So that's what we're leading up to. So van der Waals interactions, you may have also heard it called London dispersion forces. Same thing. Very, very weak. And it's just, this is what you would see to describe the interactions between nonpolar molecules. So when you have a molecule, remember that all of those electrons are not static. Okay? You've got, so let's say that we're talking about something, with a carbon. You don't think about a carbon as being charged or a hydrogen being charged in a bond with carbon and hydrogen. But the electrons are moving around. And every once in a while, you'll get a little partial positive here on the hydrogen and a partial negative on the carbon. And when that happens, then you can have a transient dipole moment where another molecule that just happens to be in the vicinity of the correct orientation gets attracted to the carbon, let's say. So it could be another hydrogen that's partial positive. That's a transient dipole. And there's a very weak, fleeting interaction. So that's all van der Waals or London dispersion forces are. And that's really all that I want you to understand. They're very weak. And just like with the hydrogen bonding, the distance between the atoms is very big in comparison to a covalent bond. Table 2.4 is going to be your friend. It summarizes the different interactions that you can have between molecules. The non-covalent interactions are much, much weaker than the covalent ones. So covalent bonds, again, that is within the molecule itself. So think about the bond between the oxygen and the hydrogen within a single water molecule. The non-covalent interactions that we're talking about are between two water molecules, let's say. And they're always forming and breaking. But why are these interactions so important? They're important because the sum of their parts can actually stabilize a structure. So right now we're looking at a crystal structure of hemoglobin. And in red, I'm going to get fancy, y'all. In red, you've got bound water. So these waters are so tightly bound that they are in the crystal structure. They're ordered, they're hanging out, they're like, hey, look, we have a place here. Without them, you can actually see the heme group here. They're both buried. We're going to talk about hemoglobin a bit down the line because it's a great way to look at a lot of different ways that proteins can interact and all kinds of fun. But the takeaway here for now is that the cumulative effect of all of these waters binding and unbinding, you have this structure that is super stable. And that's due to all of those teeny tiny little interactions. We're going to cover one more thing before we switch to a slightly different topic. We have to talk a little bit about osmosis and osmolarity. So osmolarity, I don't want you to get caught up in all the jargon. But really what we're talking about when we talk about osmolarity is the total solute concentration. Your textbook has a little bit more jargony description of what osmolarity is. Don't worry about that. It's the total solute concentration. Isotonic solutions. That means that if you have a cell in solution, that solution has the same concentration of solute as the cell does. So there's no net water movement. Remember that the cell membrane is semi-permeable. So you can have movement of ions and things across the membrane, and you can have movement of water across the membrane. And the cell is going to want to equalize its environment. So isotonic, no net movement. Hypertonic means that the cell is in a solution that is more concentrated. So the cell is going to try to release water, and it's going to shrink while doing so. That's very, very bad. And this is called crenation. When we're talking about like red blood cells, they'll just keep giving out that water and they'll just shrink and shrink and shrink. You could also have the opposite, a hypotonic solution, where you have a cell in a solution and that solution has a lower osmolarity than the contents of the cell. And in that case, water is going to move into the cell to try to dilute down the contents. And that cell will swell up and it could eventually burst. When it comes to red blood cells, that is called hemolysis. And we're going to talk a little bit more about the relevance of that in class. But I just want to throw that in here now so that we can talk about it later. Now we're going to talk about the ionization of water, and we're going to go over weak acids and weak bases. If this is a review for you, then you are in great shape. If it's not a review for you, that's all right. So water is slightly ionized. If you have pure water, you're going to have a tiny, tiny bit of hydrogen atoms and hydroxide atoms. And by atoms, I mean ions. So this is the hydroxide. This is hydrogen. Those hydrogen ions are hydrated immediately. And what that means is that it's immediately attracted to another water molecule. And it forms what's called hydronium. This reaction is, well, it's not really a reaction. It's ionization, and it's reversible. So you'll see these arrows, one going forward and one going backwards. That means it's reversible. you're constantly forming hydronium, and then you're making water, forming it, going back and forth, back and forth. We can talk about this using an equilibrium constant. We'll look at a generalized reaction first. We have A plus B, and we're going to yield C and D. We've got our reactants on the left-hand side, products on the other. The equilibrium constant, or the KEQ, is defined by the concentration of the products over the concentration of the reactants. This may look a little familiar to you from general chemistry again. And what we look at here is the product of the concentrations. So we care about the concentration. Always, always, always. And what you can determine from this is, let's say that you have a really large KEQ. Then that means that your reaction favors the products. The products are in the top, right? So if you have a really big number, then that means that the numerator must be really big, so the concentration of products is large, favors the products. If you have a small equilibrium constant, then that means that you're going to favor the reactants. So just think about fractions. A really big denominator means that you're going to have a really teeny tiny KEQ. This may bring back memories of Le Chatelier's principle and things like that from general chemistry. If you're trying to be like, I feel like I've seen this before, it's probably from that. Now let's bring that forward to water. We want an equilibrium constant for the ionization of water, which is reversible. Remember our equation. Products are on top. The only reactant is the water, which is on the left, and that's on the bottom. To get the concentration of water, it's the density of the water over the molecular weight, which the molecular weight is the same thing as saying the molar mass. Biologists like molecular weight, chemists like molar mass. They're the same thing. You can calculate that and then substitute it into your equation. So instead of having the brackets with H2O, we now have water in there. The Kw, which we will be using, is the ion product of the concentration of the hydrogen ions and the hydroxide ions, and it's at 25 degrees Celsius. If you substitute in the equilibrium constant, then you can actually calculate the Kw. So to write it out fully, and you don't always see units with the Kw. Your book does use units. So that Kw is going to be very helpful when it comes to doing problems with pH, which will be coming up. I'm sure that you've encountered the pH scale by now. This is a 300 level biochemistry course. You know what pH is. But just as a refresher, The pH scale is based on the ion product of water. And to define pH, it's the negative log of the concentration of hydrogen ions. If you have a neutral solution, you've got a pH of 7. We will be using this equation, so it's something that you should add to your memory. This little diagram just shows you the pH of some fluids to give you an idea of where they fall. So gastric juices, all those, the stomach acid, very, very acidic. If you're a coffee drinker, it's a little acidic. We've got some human blood, tears, that's very, very, very slightly basic. If you use bleach when you're washing your towels, that is incredibly basic. Now, why do we care about pH? Well, I mentioned that human blood is very, very slightly basic. It's got a pH of around 7.4. Typically speaking, the pH of your blood is kept very tightly between 7.35 and like 7.45, somewhere in there. If the pH of your blood dips below that, so it is smaller than 7.35, that is called acidosis. And it can occur in folks who have severe uncontrolled diabetes. We're going to address that kind of at the end and bring a bunch of concepts together about why that happens. And by the end of the course, you'll be able to talk about that intelligently because you'll know a lot of the metabolic pathways and the underlying reasons for why this happens. You can also have alkalosis, which is having a pH that's above 7.4, and that, again, life-threatening, okay? If the pH of your blood is wrong, then you're going to have all kinds of bad things with your red blood cells, where they're shrinking or they're swelling and all kinds of badness. So you do not want to mess with the pH of your blood or any other bodily fluids for that matter. So no raw water. In all of this talk of pH, we have to talk about weak acids and weak bases. Let's introduce some terminology. Conjugate acid-base pair. You've got a proton donor, proton acceptor. What you'll see as the generic equation where HA is your acid, it is also the proton donor. And the A- is your conjugate base. So when that acid dissociates, it forms a negative ion, and that negatively charged ion is the conjugate base. The stronger the acid, the greater its tendency to lose its proton. So please remember that. This chart, don't worry about memorizing it. It just gives you a visual for some different conjugate acid-base pairs and some of the different acids that you'll see. We'll be talking about acetic acid. We'll also be talking about carbonic acid a little bit later and phosphoric acid, just to give you a heads up. You don't need to memorize any of these values. Just take it in, enjoy it for what it is. When we're talking about equilibrium constants with water, we can move that same concept forward to talk about acids and bases, but we're just going to focus on acids here. That same little generic equation, that's what we're going to use to define our terms. We have an equilibrium constant for this reversible reaction. We're going to take our products, put those at the top, the numerator, the reactant, the acid, goes on the bottom. And this KEQ, we also call acid dissociation constant. So when we're talking about weak acids, you'll see Ka, and that means acid dissociation constant. Just like we talk about the hydrogen concentration and the negative log of that gives us a pH, the negative log of the Ka gives us the pKa. When we talk about proteins and we talk about amino acids, we talk about enzymes and how they work, enzyme mechanisms, the pKa is going to be very important. So understanding what a pKa is, you need to hold on to that concept. The stronger the tendency to dissociate a proton, the stronger the acid, right? So if you have a really strong acid, it's going to let go of that proton easily, willy-nilly. When that happens, what you see is a low pKa. So let's write this out. I am a visual person, so I like short little bursts of text or pictures that help me remember things and solidify concepts. Strong acid. What does that mean? It's got a low pH, low pKa. So if I gave you a list of acids and I told you their pKa's, you should be able to order them from the weakest acid to the strongest acid, simply because you know the pKa. You can determine the pKa experimentally, and we'll talk about that now. The way you do that is by creating a titration curve, and that's a plot of the pH of your solution against the amount of sodium hydroxide acid added, which is a strong base. This is an experiment that you might do in a biochemistry lab class. So let's say that you've got a flask here and it's got a known concentration of an acid that you care about. Let's say that it has an amino acid in it. It's got glycine. Glycine has carboxylic acid, and we can do a titration to figure out the pKa of this amino acid. So you're going to titrate in sodium hydroxide a little bit at a time, stir it, and then take the pH. And you keep repeating that. So you may have glycine that's at a pH of, say, about 2. You're going to keep repeating this, add a little bit of sodium hydroxide, stir, take the pH, until the pH of your solution is like 10 or 11. And all along the way, as you're taking those measurements, you can plot the equivalence of sodium hydroxide. What we're really talking about is how many moles of sodium hydroxide have you added. And you plot that against the pH of the solution. And you'll get some kind of a titration curve. Now, I'm not saying that represents what it will look like for glycine, but you can get some kind of a general sort of sigmoidal shape for a titration curve. And thankfully, we have a better figure to look at in terms of the, the numbers and things. I am not an artist, but I try. We talked about the pKa and that you can derive that from a titration curve. Well, how do you do that? At the point where you have equal parts of protonated and deprotonated acid, and in this case we're looking at acetic acid, protonated, deprotonated. At the point in time where you have equal parts, that is your pKa. So this midpoint here, you follow it up, okay, I've got 0.5, and then you take that over to look at the pH of your solution. The pH of the solution, when you have equal parts of protonated and deprotonated acid, that is the pKa. Now you'll see that this flat region is kind of highlighted in blue. We're going to talk about that next. It's called the buffering region. So that leads us to buffers. Before we talk about that buffering region, we're going to define a couple of things first. Then we'll go back to that same titration curve and talk about it some more. So a buffer is just an aqueous system that can resist changes in pH when you add a tiny bit of acid or a tiny bit of base. The key word here, small, tiny. You can't dump, a liter of sodium hydroxide into a buffered system and expect the pH to stay the same. A buffer system consists of a weak acid and its conjugate. So let's say that we had that acetic acid, right? If we mixed acetic acid and sodium acetate, that would be a buffering system. It's pretty much the salt of the conjugate base. So if you find a salt that has that conjugate base as a part of it, you dissolve that in water, there you go. Now back to this buffering region. It's the flat part of a titration curve. It's not exactly flat, but it's the most flat. And the reason is, look at where we start here. So this buffering region, we'll bring that down. The majority of the sodium hydroxide that you add is added in this buffering region. But the change in pH is pretty small. So you're adding all of this base, but the change to the pH of the solution is pretty minimal. That's what a buffer is. So that zone around the pKa where the pH isn't changing very much is exactly where you want your buffer to be. So if you're trying to come up with a buffer for a system, you want to find something that has a pKa that's approximately the pH that you want your system to be. The Henderson-Hasselbalch, I can never say that word correctly. All these years. Look, some words you just know they are a formidable foe. Henderson-Hasselbalch. I struggle with that every time. So if you struggle with it, that's okay. You can still get a PhD and struggle. Anywho, this equation describes the shape of the titration curve of any weak acid. And we can also use it to calculate things. So the pH of your solution is going to be equal to the pKa plus the log of the ratio of your conjugate base over the acid. So let's think about this. If I have equal concentrations of the acid and the conjugate base, then what that means for my equation, the log of 1 is 0. So when I have equal concentrations of conjugate base and acid, then my pH is equal to my pKa. When you're making a buffer, that's the ideal makeup. So equal parts acid and conjugate base. So if I give you some concentrations, you should be able to determine what the pH is if you know the pKa. And you should also be able to look at a list of, 3 or 4 different buffer systems and choose which one would be the best buffer for a particular region based on the ratio of the conjugate base to the conjugate acid. So the closer you are to 1, the better the buffering capacity you have. So this graphic just shows kind of how a buffering system works. You've got acetic acid, you've got some extra acetate added as well. If you add in some base, then that base is going to pick up a hydrogen from the acetic acid and you make water. Let's say that you add in some acid. Well, that is going to be attracted to that acetate ion and you're going to end up making more acetic acid. So you need to be able to think through how the buffer system works. What happens if I add acid? What happens if I add base? Now, inside of your cells, you've got all these different macromolecules, and they are made of amino acids, they're made of nucleotides, and all these things that have the ability to buffer against pH changes. One example is histidine. Histidine is an amino acid, and it has the potential to be protonated or deprotonated. It's got a pKa. The side chain has a pKa of about 6. So that means when the pH is lower than that, you're going to have a proton. When it's higher, it's going to be deprotonated. So it buffers effectively around the same pH that, your blood and tissues and all those things are at, about 7. So you've got all of these different little ways that your body protects against changes in pH. In the cytoplasm, phosphates is essentially a phosphate buffer system. Phosphate, so phosphoric acid is a triprotic acid. Triprotic means that it's got 3 protons. In the cell, what we're looking at here, we've lost one of those hydrogens to make a hydrogen atom and a conjugate base. This buffering system is effective at its close to its pKa. 6.86 is the pKa, so it works over a pretty broad range, 5.9 to 7.9. That means that it will help to keep your body happy, keep it around the right pH. In the blood, you've got a different buffering system. It's the bicarbonate buffering system. I promised you we would talk about these different acids, and here we are. So you've got carbonic acid, when it loses a hydrogen, you make a hydrogen ion and bicarbonate. That's what this is, bicarbonate. You can write out the equilibrium for this, where you put the acid on the bottom, the products on the top. Why do we care about that? Because there's more to the story. The way that we form carbonic acid is from the carbon dioxide that we're breathing in that's dissolving into our blood. Okay. So remember, when you're respirating, the technical term, you're taking in all the gases that are in the air. So oxygen, nitrogen, carbon dioxide, all of that is going into your lungs. When you exhale, you're breathing out a lot of carbon dioxide, nitrogen, and you're breathing out some oxygen too. We don't take it all in. But some of those gases are dissolved in the blood. And that dissolved carbon dioxide is what makes that carbonic acid. And remember from earlier, nonpolar gases don't really like to be dissolved. So we're not making, a ton of carbonic acid from just breathing, but we are making some. There are other ways that our body kind of helps to produce and maintain that buffering system that we're not going to talk about. The pH of the bicarbonate buffer system exposed to a gas phase depends on the concentration of bicarbonate and the partial pressure of carbon dioxide, which is the concentration of the carbon dioxide in the gas phase. Okay. Partial pressure, that might ring a bell from talking about gas laws and things like that in general chemistry. But those are the things that help govern the pH of that system. And thankfully, that system is effective around 7.4, which is what our blood needs to be at. You can change the pH of your blood simply by breathing differently. Which, isn't that crazy? Simply breathing, something that is an involuntary action controlled by the brainstem, you can adjust the pH of your blood. So if you hyperventilate, which means you're breathing rapidly, that will raise your pH because you're taking in a lot of carbon dioxide and you're breathing out a ton, right? So you're just by doing that, you're changing the partial pressure of carbon dioxide and adjusting the blood pH. Something so small, right? So we talked a lot about pH, and the reason is it matters quite a bit. All of the proteins and enzymes in your body optimally work at the pH of your body. So 7.4 for most things. But there are some exceptions. For example, pepsin. Pepsin is a protease. Protease is just something that eats proteins, right? It breaks them up. When I say eats, I really mean that it does a chemical reaction that will break apart a protein into smaller peptides. But eats just sounds a lot better. So it's a protease that works in the stomach. And what do we know about the stomach? It's a very acidic environment. So optimally, its pH is very low, somewhere between 1 and 2. That's where it has the maximum catalytic activity. However, you look at another protease, like trypsin. That's a protease that's in your small intestine. Its optimal pH is about 7-ish. So your body needs to maintain the pH very tightly so that all the enzymes can do their work and your proteins stay folded properly and interacting with the right things. If your pH gets, too acidic or too basic, that's absolutely life-threatening because all of the things in your body don't function well. And that leads us back to talking about untreated diabetes and acidosis. So when you're diabetic and you don't know it, you have really high blood sugar. Your body doesn't recognize, hey, there's a lot of glucose in my blood. I should take it in and make energy. Instead, your body constantly thinks that it's in starvation, and it will use stored fatty acids to create the energy that your body needs. And in doing so, you're generating a lot of acidic metabolites. And those metabolites include beta hydroxybutyric acid and acetoacetic acid. Acid and more acid. So you're increasing the amount of acid that's in your blood, which results in a decrease in the pH. Remember, buffering systems are not perfect. They can buffer against small changes, but big changes are a big problem. That results in acidosis. And if it's not caught in time, then your enzymes are not able to function properly. And it is life-threatening. So, with that, thanks for watching. That is Chapter 2. Make sure that you tune in for the live lecture, Tuesdays and Thursdays, where we'll focus more on practice problems. You can get your questions answered, and we'll do activities to help prepare you for the exams and give you some more information about how all these concepts are related to life and your own health. Again, thanks for watching, and stay safe.